Okay, well, it's noon. I will get us started now. First, a few housekeeping notes. I really want to thank Claudia Steele O'Sullivan again for organizing this seminar series. Fantastic job. Thank you, Claudia. And just to remind all of the participants to please post your questions into the Q&A tab. And then at the end of the talk, I'll be able to ask those questions. And also, please remember that these talks are recorded, so please check out the website that you see on the screen right now if you want to check out any of the recorded talks. So with that, it's my great pleasure to introduce a mentor, a colleague, and a friend, Dr. Morag Park. Dr. Park is originally from Scotland, and she's been at McGill University now for a number of years and really launched her career there. She serves as the director of the Goodman Cancer Research Center at McGill, and she's also a James McGill professor and holds the Diane and Salguerra Chair in Cancer Genetics at McGill. So Dr. Park has long thought about heterotypic interactions and the crosstalk between tumor and stroma, particularly with expertise in HGF signaling through the MET receptor, which she cloned. And from there, her work has evolved into a really deep understanding about tumor and microenvironment and spatial relationships that are, now clinically relevant and directing our understanding about responses to therapy. And I just want to mention a landmark paper that she published that with first author Greg Phenak in 2008 that really was the first to molecularly profile the tumor microenvironment. When everyone else was microdissecting out tumor, they microdissected out stroma and identified signatures that are clinically relevant and predict outcomes. So, that landmark work really inspired all of us in the microenvironment field, and it's where we are today because of her. So without further ado, it's again a sincere pleasure to introduce Dr. Morag Park. The screen is yours.
Okay, so my computer is just making odd noises because it's heating up, so let's hope it keeps going. So thanks very much, Sandra, and I'd like to thank you for the invite to give this talk. And I'll see if I can get the slides working, and you maybe tell me if everything is going okay. So does this look okay?
Looks great.
It looks fine.
Okay, perfect. So I decided I would give some background on some of our older work and then some updates on where we're going now. And I think particularly, as Sandra said, we started being interested in the stroma, particularly in breast cancer and more recently in triple negative breast cancer. But we're now also interested in how the tumor itself and the tumor cells are organized within the tumor. So we're beginning to ask questions about both of these microenvironments now. And we're starting to ignore stroma and focus on the tumor, believe it or not. So let's see if I can go forward. Okay, so I don't think I need to tell this audience that triple negative breast cancer is a heterogeneous disease and remains a therapeutic challenge. It really is the first cancer that was stratified really for personalized medicine or precision medicine type therapies, in as much as we can identify readily, I think, in every clinic, estrogen receptor positive patients and treat them accordingly with tamoxifen, for example, HER2 amplified patients. But this subgroup that are called triple negative because they're negative for the estrogen receptor, HER2, and progesterone receptor are still a clinical challenge. Traditionally, or in most centers, particularly in Canada anyway, in a socialized health system, these patients are still stratified for nowadays neoadjuvant chemotherapy. But what we know from many studies is that within the triple negative subgroup, there is a subset of patients that respond very well to chemotherapy and a subset of patients that actually have poor outcome. And the challenge, of course, is to identify between these two distinct subtypes as a mechanism to stratify these patients and obviously consider to change the therapeutic options. Together with our understanding of the disease, obviously the large sequencing studies have started to give us some understanding. But clearly, as we all know, triple-negative breast cancer is a heterogeneous disease, and there are multiple pathways that are targeted. And although these are potential therapeutic targets, these therapies are only coming into the clinic really at the moment, with the exception perhaps of the BRCA2 and BRCA1 DNA repair targets. But this stratifies triple negative with mutations in the PI3 kinase mTOR pathway, a subset in the RAS pathway, different regulators of the cell cycle, but as well as some growth factor receptors. This includes the receptor that I've spent much of my life working on, the MAT receptor, but also FGF receptors that are more and more being identified as amplified. But again, these genomic markers are still difficult to stratify patients into those that are predicted for good versus poor response to neoadjuvant chemotherapy. Okay. So we had, as Sandra indicated many years ago, essentially 15 years ago, started to consider really what else is involved in breast cancers that would influence outcome. And I really won't go through this, but we used back then laser capture microdissection, this on the Agilent platform because the Affymetrix platform wouldn't even support it at that time. But I think some of the surprising results was that we started to see really distinct differences between obviously tumor epithelium and normal epithelium, but also between normal stroma and tumor stroma. And within the tumor stroma, we saw within the different subtypes of breast cancer that the stroma was different. This led us to obviously do a number of bioinformatic analysis, and we generated, as Sandra said, signatures that were associated with outcome. And this initially was pan breast cancer, but now we focus more on triple negative breast cancer. So for example, those signatures derived from the stroma. So stroma we were using very loosely as cells that were not tumor cells. So of course, this would include the mixture of immune cells, fibroblasts. We're clearly capturing some vasculature as well. And we can't at that stage distinguish between them. But the signatures that were associated with poor outcome, particularly signatures of fibrosis, cancer-associated fibroblasts, as well as matrix remodeling. And signatures with good outcome tended to be signatures that were associated with immune content. We could stratify those signatures across all breast cancer subtypes. And this is just to highlight that, more than 12 years ago, we already could see that immune signatures were particularly strongly associated with good outcome in the basal subtype of triple negative breast cancer, so the molecular basal subtype of triple negative breast cancer, as well as the HER2 amplified breast cancers. But were less correlated with outcome in either the luminal A subtype or the luminal B subtype, for example. In addition, the signatures that I mentioned, for example, matrix remodeling was associated with poor outcome in basal breast cancer, and hypoxia was particularly associated with poor outcome in HER2 breast cancers, looking at the gene expression anyway. So the inference of these immune signatures got us interested, obviously, in looking at the immune landscape. And as we now realize, there's a lot of interest, particularly in tumor progression, whereby as tumors progress, they are now recognized or not by the immune system, such that tumors that do evolve and progress undergo multiple mechanisms whereby they evade recognition by the immune response. And this can be through multiple different mechanisms, either through immunoediting, immunosuppression of immune cells that are recruited into the microenvironment, and of course now the therapies such as the checkpoint therapies that are prevalent in the clinic and even in a socialized healthcare system in Canada, such that the progression of cancers, and breast cancer included, is associated with mechanisms of escape from immune recognition. Because we had these gene expression signatures, we felt it was important to use these to begin to ask, can we actually predict outcome in large data sets? So this is over, I think it's 5,000 different breast cancers that were collected within this data set. And what was recognized was that if you use immune signatures, you can predict the good outcome patients, such that the immune signature would predict good outcome, but also immune signatures would still be associated with bad outcome. And so basically what that indicated was that there was a bad outcome subset of patients that had an immune signature that were very difficult to predict. And of course that raised the question, what was the difference between those patients that were easy to predict by a very simple immune gene expression signature versus those that were hard to predict? So we decided at that time to go back into our data sets and start to look at that in more detail. And what was very important at that time to us was the publication of this paper. So this is a large data set of patients, over 12,000 breast cancer patients with outcome. And in this is really a consortium of pathologists led by Torsten. To be honest, I don't think Torsten Nielsen was the last author. I think it's Carlos Caldas was the last author in this paper, who really assembled a large data set of breast cancer slides where they annotated the location of CD8 T cell infiltration. And in this diagram, what they've highlighted in, I think it's nearly 2,500 patients, is the ER negative subset. So that's essentially those patients that we saw immune signatures being associated with outcome in. So if they annotate these patients, what they can show is that if there are no CD8s at all, that's associated with the worst outcome. The presence of CD8s in the yellow line in the tumor and the stroma, that's associated with the best outcome. But in this data set, what they showed, I think probably at large scale for the first time, was that the location of CD8s only in the stroma was associated with outcome which was nearly as bad as patients that had no CD8s at all. Since then, there's been many studies that have recognized that tumor infiltrating lymphocytes, just like the signatures we could see, were more frequent in the triple negative subset and the HER2 subset of breast cancer. They are now associated with an increased pathological response rate to neoadjuvant chemotherapy, for example, improved outcomes even in unselected patients. And because of this publication and some of the work that we've contributed, there's now increasing efforts to standardize the assessment of tumor infiltrating lymphocytes. And the reason that this is important is that pathologists were first, the guidelines for pathologists was to actually annotate TILs in the stroma. So the understanding that TILs in the stroma may be associated with poor outcome led to the need to understand better what the difference was. So we decided at that time, through the publication of that paper, to really start to go back in and look at the patients that we had derived gene expression signatures by looking at stroma and epithelia. And for this, we used a cohort of triple negative breast cancers where we had previously subjected these to laser capture microdissection. And we've got 50 triple negative breast cancers in this cohort. Most of these are the basal subtype, and what we focused on for these analyses is the basal subtype. But what that demonstrated to us when we just simply stained the H&E slides with CD8 to identify CD8 T cells, that we could see three major distinct subclasses. One subclass we call fully inflamed, where the CD8s are evenly distributed throughout either the tumor bed or even at the margin. These black lines depict the margin of the tumor with the peritumoral space. A second major subset identified patients that are called now immune cold, essentially no CD8s within the tumor bed. But we saw that the majority of these actually did have CD8s at the margin. And you can see here there's a large number of immune infiltrates, and these are not all CD8s, but predominantly CD8s. So they enter towards the tumor, but very few of these CD8s actually enter into the tumor bed. The third subset are patients where the CD8s are predominantly in the stroma. So this is exactly what this large data set indicated. And you can see quite clearly, I think, that very few of these CD8s are located within the tumor nests themselves. So in a similar manner to the large data set, we could show in our data set that the cold tumors were associated with poor outcome in response to either neoadjuvant or adjuvant chemotherapy. The fully inflamed, good outcome. And those where the CD8s are in the stroma have an intermediate bit towards poor outcome. So this really highlighted a very simple scheme whereby you could actually stratify patients with triple negative breast cancer in the clinic based just on the localization of CD8 T cells. So that we can split first by CD8 low into this immune cold or margin restricted versus CD8 high. The CD8 high can be split again into either stromally restricted or the fully inflamed subset. Because we'd already generated the gene expression profiles of these patients in 2006 or something, we could then go back in and annotate and look at the different signatures. Except this time what we did is rather than allow the patients to cluster independently, we actually subdivided each of the clusters into patients that were either fully inflamed, the stromally restricted, or these immune cold subsets. And we can look at either stroma or epithelia, gene expression, epithelia being the tumor. And I think you can see quite clearly that there's distinct gene expression signatures or pathways in patients that are immune cold versus the fully inflamed, which are the red patients, or the stromally restricted. And similarly, if we look at the epithelia, we can easily distinguish three distinct subsets of triple negative breast cancer based on different gene expression profiles. So this really indicated to us that it's likely that, and I think an important thing, well, I think as you can see from the number of patients here, we essentially have about 1/3 of the patients are in the immune cold. That actually goes up to maybe 40-45%, about 1/3 fully inflamed, and about 1/3 in the stroma. Now this becomes quite important because if you remember what I told you is about 40% of triple negative breast cancer patients have a good outcome in response to chemotherapy. And what we can show here by just the localization of the tails, the CD8s, it's these fully inflamed patients that are associated with a good outcome. So by beginning to characterize the differences at the level of gene expression, going back into the patient's tissue, we were able to identify distinct biological properties in these three distinct subclasses of triple negative breast cancer, defined by the location of the CD8 T cells. So if we just do a first split in, say, immune cold versus CD8 positive, the immune cold patients were highly enriched for signatures of TGF-beta. This is this green light patients here, when compared to the stromally restricted or the fully inflamed. Associated with TGF-beta signatures, we see many markers of cancer-associated fibroblasts are elevated in these immune cold patients. And this is in particular lysoloxylase and lysoloxylase L2. There's just been a nice paper from Val Weaver's lab and other labs really demonstrating the association of elevated LOX and LOXL2 with poor outcome in breast cancer. This also includes other genes that are associated with activation of fibroblasts. Working with Roberto Salgado and Gert van der Eyden, who are the head of the International TILS working group, they re-annotated all of our slides and also annotated the association, for example, of fibrosis. So from a pathological perspective, many pathologists will annotate fibrotic foci. That's what's shown here. This would be this, what I didn't say is we always actually look at a whole tissue section rather than a punch. So for example, if you were making a TMA, this punch would be coming from the fibrotic region, this punch would be from the tumor region. But unless you took care on really annotating where your punches came from, you wouldn't really know where you were looking. So everything was scored on a whole tissue section, and both Gert and Roberto annotated the fibrotic nature of these tumors. And I think you can see quite clearly here that those that are CD8 low, that is immune cold, have elevated areas of fibrotic foci or elevated numbers of fibrotic foci compared to those tumors that are CD8 high. So this really correlates with what was really coming out in the literature through an understanding of activated fibroblasts and how activated fibroblasts will regulate an immune microenvironment. And that the activation of fibroblasts in multiple tumor types are associated with these highly fibrotic tumors. And that in this context, many of the genes that we could identify in our immune cold subset, such as the TGF-beta, many TGF-beta pathways, and interleukins such as interleukin-10 produced by cancer-associated fibroblasts, are already known to be suppressant factors, for example, for recruitment and activation of CD8 T cells. We had other cytokines and chemokines that were associated, for example, with recruitment of MDSCs, for example. Also, the transition of macrophages from an M1 anti-tumor type macrophage to an M2 type, particularly interleukin-6. So this raised the question to us, what really were we able to recapture this type of phenotype in our sections? So if we look, for example, just at macrophages, and we ask if we compare between a predicted M1 type versus an M2 type, what we can see, and for this we use two markers, CD68, that will identify all macrophages, M1 type as well as M2, versus CD206, which is dually expressed with CD68 in those macrophages that will be polarized towards M2 type, such that the M2 type of macrophages are considered to be more in a tumor promoting type capacity, whereas the M1 type macrophages are considered to be more in a tumor prevention. What we can see quite clearly, and I think you can see it here, and we have quantified this, is that the fully inflamed tumors are predominantly infiltrated by macrophages that have an predominantly M1 type phenotype, as depicted here by single positivity for CD68. As predicted, the immune cold tumors are predominantly infiltrated by macrophages that are doubly positive for CD68 and 206. So these would be of the M2 type subtype. What's also obvious in these slides, I think, is that in the stromally restricted patients, the macrophages are also predominantly of these dually positive subsets of macrophages. And again, these macrophages are predominantly localized within the stroma in these patients, whereas the M1 type macrophages in the fully inflamed patients seem to be interspersed throughout the tumor bed. So this identifies that these distinct subsets of triple negative breast cancers, annotated just by the localization of the CD8 T cells, are clearly quite distinct immune microenvironments. So we can go back into our gene expression data and we can compare now if we take the CD8 high subsets of patients, so that's about 60% of the patients, and we compare the CD8s that are in the stroma versus the CD8 that's in the stroma pathways versus the CD8s that are fully infiltrated throughout the tumor bed. I think as predicted, what we see is those that are fully infiltrated, we see activation of typical pathways for immune activation, such as JAK-STAT pathways, the interferon signaling pathways, whereas we see quite distinct pathways that are activated in the stromally restricted patients. So again, highlighting that these are really distinct subsets of triple negative breast cancer. One important question that one has to ask is we already know that CD8 T cells, to kill a tumor cell, have to come in contact with that tumor cell. They have to recognize MHC class 1 on the surface of the tumor cell through the T cell receptor, and then once activated, they will then release, for example, granzyme or perforins that will act to target the tumor cells in the vicinity of these CD8 T cells. So an important question to ask for us at that time was, are the CD8s that are recruited into this immune-activated gene expression landscape actually activated? So here we just stain for CD8 and for granzyme B and pan-CK to identify the tumor cells, for example. And I think you can see quite clearly in this slide, it seems that the quantification is missing, but we have that in our publication, that the CD8 T cells that are uniformly distributed between the stroma and the tumor bed are predominantly granzyme B positive. And in contrast, if we look at those patients where the CD8s are located in the stroma, and sorry, the color code is different here, but pan-CK is in the red, so these are your tumor cells. Granzyme B, CD8s are now in this orange color. I think you can see quite clearly that the CD8s in the stroma, very few of them, if any, overlap with granzyme B. So these CD8s really have a profile of either being exhausted or inhibited, for example. So if we go back and we look at the pathways that are elevated in those patients where the CD8s are restricted in the stroma, and ask, what's really different between these patients versus these fully inflamed patients. If we look at, for example, immune type signaling, one of the immune signals that we can identify in this subset of patients is a signature for interleukin 17A that's identified in psoriasis, but it's really a sort of inflammatory signal under conditions of psoriasis. And we were particularly interested at that time in a paper from Karin Visser's lab studying the polyoma middle T model, but where she had really been able to dissect the impact of different immune and cytokine signaling on tumor progression. And in this paper, what she demonstrated was that the release of interleukin 1 beta would stimulate the preponderance and transition of gamma delta T cells to secrete interleukin 17. This would then, possibly through cancer-associated fibroblasts, promote recruitment of and activation of neutrophils, and that neutrophil recruitment into this microenvironment would act in a suppressive manner to suppress CD8 T cells. So this raised the question, was the stromal microenvironment enriched, for example, with these immunosuppressive gamma delta T cells or interleukin secreting T cells? So here we're comparing the fully inflamed tissues. Again, in this time, the tumor cells with PAN-CK are purple. And we are looking at interleukin 17 secreting cells. And I think you can see quite clearly that there are interleukin 17 secreting cells in the stroma of the stromally, where the CD8 T cells are enriched in the stroma, and that there's essentially no interleukin 17 positive cells in these fully inflamed patients. If we look at neutrophil recruitment, we can see quite clearly again that the fully inflamed patients show very low levels or no evidence of neutrophil recruitment, whereas the stromally restricted patients show positivity for neutrophil recruitment. These are predominantly in the stroma, but they do then begin to infiltrate into the tumor beds. So again, these two subsets of patients that would be difficult to identify by pan-gene expression signatures can be stratified based on both the CD8 localization, but also by gene expression. Other pathways that were clearly distinct between these stromally restricted patients versus the fully inflamed patients were pathways of cholesterol biosynthesis and the mevanolate pathway, which is associated with pathways of cholesterol biosynthesis. We could see this both at gene expression levels of the enzymes that are part of the cholesterol biosynthesis pathway. And what's also known and what's very interesting is that in response to interferon, particularly interferon gamma, the cholesterol biosynthesis pathway is actually suppressed. So that's consistent with the fully inflamed patients having a strong interferon gamma signature and suppression, or type 1 interferon rather, and interferon gamma signature, and suppression of this cholesterol biosynthetic pathway. And perhaps, we don't know if this is elevated in the stromally restricted patients, but what this demonstrates is that this pathway is not suppressed. So again, what that would indicate is that this pathway, that these stromally restricted patients do not appear to be under the influence of type 1 interferon signaling that would act to suppress the cholesterol biosynthetic pathway or interferon gamma signaling. This is also interesting in that there's a number of papers published demonstrating that following viral infection, particularly in macrophages, you will also, in response to type 1 interferon, suppress the cholesterol biosynthesis pathway. So this is consistent again with a microenvironment that's enriched for type 1 interferon. In addition, a number of papers have come out recently, and this is from 2019, demonstrating that cholesterol in the tumor microenvironment will also induce CD8 T cell exhaustion, and that this is regulated in part through an ER stress XBP1 dependent manner, whereby cholesterol in the microenvironment that's uptaken by tumor cells and immune cells will promote an ER stress response and expression in particular of checkpoint or inhibitory receptors in immune cells, decreasing the amount of granzyme B produced by the CD8 T cells, correlating with an exhausted phenotype in comparison to cells that have a low level of cholesterol in the microenvironment. So this is entirely consistent with what we're seeing in the gene expression signatures that we find in our stromally restricted patients. So essentially what we can now do is propose a subtyping scheme based first of all on the location of CD8 T cells, where we either have immune low or immune cold versus immune high. Signatures of fibrosis high in the immune low versus fibrosis low. So we can split patients by gene expression signature. These define the margin restricted patients. So if we then use these signatures to annotate a distinct data set, and this I realize isn't on my slide. Oh, here we go. This is the RODI data set, which has over 500 triple negative breast cancer patients. If we first split by immune high, fibrosis low, we get good outcome. Immune low, fibrosis high, we get poor outcome. So this is essentially the type of split that we usually see for outcome in triple negative breast cancer. But as we know, the stromal localization identifies an additional cohort of patients that appears to have an intermediate outcome, at least as seen from this large study by Carlos Caldas and Torsten Nielsen. So we can then take these immune high patients, split them again based on the interferon levels. So interferon low, this is the stroma restricted, versus interferon high, the fully inflamed. Then we can use, in addition, the cholesterol signature, cholesterol high for the stromally restricted, cholesterol low for the fully inflamed. And we can apply this split to the purple line, which is the CD8 positive patients. And what we can see in this additional data set, the RODI data set, that we can now identify these three subtypes of patients. Those that are immune cold with the worst outcome, that have the signatures that we identify in the strongly restricted with an intermediate outcome, but towards poor, and those patients that have this fully inflamed signatures, which have the best outcome. However, as I showed you, these outcomes in these patients can easily be stratified just by the localization of the CD8 T cells. So this localization, as I've told you, is now actually really championed by Roberto Salgado and Bert van der Eyden, who are the head of the International TILS Working Group, have now developed new guidelines for pathologists to both count CD8 T cells, but to annotate their localization in the stroma versus throughout the tumor, such as these fully inflamed patients. But what this now shows us is that we can now identify really distinct subsets of triple negative breast cancer patients based on the location of the CD8s. What I've shown you is that the immune cold or margin restricted are those highly fibrotic, CAF activated with M2-like macrophages. The fully inflamed tumors, as predicted, where we now have a lot of understanding, have signatures of type 1 interferon, epithelial granzyme B in the CD8s, and this really anti-tumorigenic M1 type polarized macrophages. The strongly restricted patients really have a distinct microenvironment that is elevated in cholesterol synthesis signatures when compared to those patients that are fully inflamed. They have predominantly stromal granzyme B, but not within CD8s, and they have this really immune inhibitory type, probably gamma delta T cells that are secreting interleukin 17 and recruiting neutrophils. Now, clearly, an important question, if we're thinking on evolution of triple negative breast cancers, the question is, are these tumors adapting? So does a fully inflamed tumor adapt to become a stromally restricted tumor? Or can a stromally restricted tumor adapt to become a margin restricted tumor? And obviously, our understanding now, particularly looking at exhaustion of CD8 T cells, is often associated and correlated with the expression of immune checkpoints or immune inhibitory molecules. So clearly, there's been a lot of interest in looking at, in particular, members of this family, particularly once activated CD8 T cells will, once in response to interferon gamma, for example, will elevate PD-L1, for example, and PD-1 in the T cell. And that this recognition will, in addition to the recognition of peptides through the T cell receptor with MHC class 1, really promote this activation of this pathway and subsequent inhibition of the T cells in that microenvironment. This can also be caused, as we know historically, by a number of oncogenic pathways that will induce, for example, in tumor cells expression of PD-L1 that itself can act also in an immunosuppressive manner to enhance immunosuppression of T cells that are expressing PD-1. So that led the question, if we want to characterize these distinct subsets of CD8 of triple negative breast cancer for the localization of PD-L1. This is a little bit busy, but if you only focus on the green staining at the moment and the blue, so blue is DAPI, green is PD-L1, fully inflamed, stromally restricted, and margin restricted. What we see is that PD-L1 is localized in the tumor cells, predominantly within the tumor nests, and we do believe in the tumor cells, in the fully inflamed tumors. PD-L1 positivity is identified in the strongly restricted, but in that context, the PD-L1 positivity is predominantly within the stroma. We have been able to identify that some of these PD-L1 positive cells are macrophages, but there are clearly additional PD-L1 positive cells in the stroma that are not M2-like macrophages anyway. And importantly, these immune cold tumors, surprisingly perhaps initially, are PD-L1 negative. If we look by gene expression, this is actually supported by the gene expression that we had identified. So PD-L1 is CD274. In the fully inflamed, we see that it's predominantly expressed in the epithelia. When we look at the stroma, it's expressed in the stroma in the fully inflamed, but also expressed in the stroma in these strongly restricted patients. What's also important is that many other immune modulatory molecules are expressed and again, predominantly in these fully inflamed patients. And one of these, so we asked really a simple question, were any of these anti-correlated with PD-L1 expression and with CD8 T cell density in the tumor core? And one of these, VTCN1, which is B7H4, which is a member of the PD-L1 family, is anti-correlated with PD-L1. So if we stain these same patients with B7H4, so PD-L1 is B7H1 of the PD-L1 family, for example. B7H4, what you can see clearly here, is highly expressed in the tumor epithelium in the immune cold patients, is expressed both at a low level in the strongly restricted patients, and some in the stroma and some in the epithelium, and is not expressed in any of the fully inflamed patients that we've annotated so far. So again, this identifies distinct subsets of patients that express distinct subsets of immunomodulatory molecules, where the fully inflamed have PD-L1 predominantly in the epithelia, the strongly restricted PD-L1 in the stroma, and the immune cold or margin restricted B7H4 predominantly in the tumor epithelia, and essentially not in any cells within the stroma. This is now consistent by a number of studies, other studies that have since been published. And there was a very nice report by Stewart et al. Using nanostring, which essentially demonstrated a very similar anti-correlation between B7H4 and a family member, B7H3, and PD-L1 expression and outcome in triple-negative breast cancer. It's important to highlight that John Rim had previously demonstrated anti-correlation between B7H4 and PD-L1 in a lung cancer cohort. So this seems to be fairly consistent between a number of solid cancer cohorts. So that, really allows us to highlight not just the location of CD8 T cells, but that these and distinct tumor microenvironments, but these distinct subsets of triple negative breast cancer are also associated with these distinct subsets and localization of these immune modulatory molecules. So again, this really begs the question to think about new types of therapies. B7H4, as I said, is a member of the PD-L1 family. It has been identified at high levels on tumor cells, and particularly papers from Genentech had previously documented this in solid tumors. The ligand for B7H4 has not been identified yet, or at least solidified. B7H4 was first identified, at least by some of the work by Pam Ohashi, as a T cell expressed in T cells, and had been correlated with immunosuppression when expressed in T cells. It's also it's also expressed in APCs. And again, clearly, this allows this development of a sort of microenvironmental network between these modulatory proteins that are expressed in tumor cells and the recruiting T cells that are recruited in the APCs that are recruited into that microenvironment. And that the joint overexpression of B7H4 and B7H3 in solid tumor types that have been studied so far are associated with these immune cold type landscapes. So whether it's really a direct suppression of T cells that are recruited into these tumors or not has really needs to be tested. Okay, so this just summarizes that in addition to these distinct microenvironments, we can now begin to annotate these tumors by these essentially markers of each subtype: epithelial B7H4, stromal PD-L1, or epithelial PD-L1. This has stimulated a number of groups to start to think about, should we really be annotating triple negative breast cancer patients and based on the localization of the CD8 T cells, and start to think about distinct mechanisms of therapy for these patients. And this is a very nice analysis by Ari Barecci in a paper in 2019. And so he used our data for our tumor microenvironment subtypes and overlaid this with the subtyping of triple negative breast cancer that had initially been annotated through Lehman et al. And Sandra Petropoil. So what this showed was that our immune cold subtype with the high fibrotic signatures essentially immune cold was an overlap of the androgen receptor subtype of Lehman et al., the mesenchymal and mesenchymal stem-like subtype. So this is overlap by gene expression profiling. These together are associated with hypoxic signatures, as I showed you, this activated CAFs, B7H4 expression, and some association with loss of STING-like signaling and beta-2 microglobulin. The immune fully inflamed, so these CD8 fully inflamed tumors correlate very well with the immunomodulatory subtype of Lehman et al. So these are likely the triple negative breast cancer patients that would respond best to different checkpoint therapies. And I think what we're seeing is that a subset of triple negative breast cancer patients do respond to checkpoint therapies. The initial studies actually did not identify that so clearly. But if you remember, these are the patients that already have a good outcome and a good response to chemotherapy. So it's likely that in the initial trials, this subset of patients were poorly represented in the cohorts, which were mostly in the metastatic setting. The stromally localized patients clearly are a challenge. These are the patients where I showed you had this higher cholesterol signaling. They do also have loss of beta-2, and we showed HLA signaling, for example, and this stromally localized PD-L1 and B7-H4. And these are the patients that are probably also difficult from a therapeutic option and may be suitable for other types of therapeutic interventions, particularly anti-B7-H4 type therapies for both of these subtypes that are coming down the pipeline. So I'd like to just change for the last few minutes of this talk, and I realize it will just be a few minutes, but just to give you some ideas of the directions that we're going. Understanding that tumor heterogeneity impacts patient outcomes, and it really highlights the therapeutic challenges, particularly for triple-negative breast cancer. So to start to begin to address that, we've actually started to use pipelines of patient-derived xenografts that we've started in the lab. In here, for many of these, we have the primary tumor in the matched metastasis that we've established as patient-derived xenografts, and we've set up pipelines like many people have done to use single-cell expression type annotation and to then remap signatures. And in this case, we're only looking at the tumor cells. So we can eliminate all of the mouse reads and really just capture the heterogeneity within the tumor. And then the important question for us is, are we identifying distinct tumor clones, or are we identifying distinct tumor biology? So to do that, what we wanted to do as we develop these gene expression subsets, we wanted to identify markers that would allow us to remap back into the tissue each of these distinct epithelial tumor cell subsets. So we've done that by identifying markers that best annotate each of the clusters. Then we develop antibodies that then allow us to go back into the PDXs, but of course also the patient's tumors from which these are derived. So by taking this approach, we've the first tumor, and we've done a few now. What I'm showing here is really 7 distinct subsets of cells that we can identify that are all human, no mouse. Different biologies. We have hypoxic subsets. We have really several that are in cell cycle transitions. And we have 2 that are quite distinct, that are more restricted. One that's enriched for basal-like markers, for example, and another that has some interferon or this sort of antiviral response. If we do a sort of monocle projection, what we can identify is that these basal subset that have signatures of bipotential cells really are at one end of the monocle trajectory, cell cycle sort of intermediate, and perhaps as predicted, the hypoxic cells with hypoxic signatures are at the other trajectory of the monocle analysis. So really, that gives us some understanding that you may transition from these cells into a proliferative state and into this hypoxic state. So we can annotate this back into the tumors. As I said, we chose genes that would best identify each of these subsets. These genes were not chosen in a selective manner. They were actually chosen by bioinformaticians that had really no clue about why we would be excited about them. But I think it's important to point out that 2 of the genes, GLUT1, the glutamine glucose receptor transporter, and CA9, were genes that were identified as associated with these hypoxic clusters. They were not chosen based on functionality, but really just on the robustness to identify each of the clusters. So we can then go back into the tumor sections using these antibodies. So on a single tumor section, we can annotate, for example, areas that are high for CA9, areas that are high for GLUT1, and the other types of markers. We can then computationally integrate the intensity of each cell, for example, for GLUT1, CA9, TOP2A, whatever marker we're looking at, and then annotate the computational clusters. But the important thing to point out here is that the patterns we see by the computational integrated clusters are very similar to the patterns that we see using the single antibodies. So this really gives us a tool to begin to map where in these tissue sections each of these subsets of cells reside. And because we can annotate them computationally, we can also measure distance from different domains in the tumor. So for example, if we look at the areas of necrosis, what we can identify is that close to the areas of necrosis, we identify the hypoxic clusters that can be annotated by CA9 or GLUT1. Interestingly, we don't have a direct overlap between GLUT1 or CA9. And I think what we believe is happening is that we know that in response to hypoxia, HIF1 becomes stabilized, and two of the targets of HIF1 are GLUT1 and CA9. GLUT1 is induced rapidly in response to hypoxia, whereas the establishment of CA9 positivity usually takes several days. So we interpret this as cells sensing a hypoxic gradient and those where the hypoxia is well established. Importantly, this changes a number of this sort of landscape. We know that uptake of glucose is associated with a glycolytic switch, and that many of the gene sets that are in these hypoxic clusters are associated with glycolysis, whereas in the non-hypoxic clusters, we see signatures of OXPHOS, for example. We can then map the proliferative signatures. One of these, PAF15 PCNA associated factor, you can see when we map it computationally that these are away from the hypoxic zones. And I think you can see quite clearly here that those cells that are positive for proliferative markers for the S phase of the cell cycle, but all of our proliferative markers are essentially anti-correlated with these hypoxic zones. So that's probably not surprising, but I do think it's quite dramatic when we observe it. What's important is that we can go back into the patient's tissue with exactly the same markers, and we can show, for example, in that patient that gave rise to this PDX, we see GLUT1 around necrotic zones, CA9. The pattern is essentially the same. KIA, this is this PCN, the S phase marker that I just told you about, is anti-correlated with GLUT1 and CA9, and that these patterns in the patient are very similar to the patterns that we see in the PDX. And we can quantify this, and we can show that the quantitation gives us a similar subset. Importantly, if we look throughout a number of PDX or patients, we see that these patterns are very similar. So there's a hypoxic zone, proliferative zones, and that these are really not differences in genomic states, but really reflect biological states of these tumors. I just want to leave you in the last couple of minutes with some thoughts about these subsets that we identified that are quite distinct. So these, we marked this with a yellow marker, it's crystalline AB, again, not associated necessarily with biology. But I think what you can see here is that these cells that are positive for crystalline AB are both within the proliferative zone as well as the hypoxic zone. What's very interesting is that those cells, as they enter into the hypoxic zone, they now begin to adapt and adopt expression of CA9. And what I told you was that the cells that are in the proliferative zone are not these cells, as they enter the hypoxic zone, stop being proliferative. So this is really an indication that these cells are likely in really undergoing plasticity. And I'll just cut to the chase here. If we isolate CA9 positive cells, these are the most tumorigenic when we re-inject them into mice. And I'll skip this. And we can actually then treat the mice both with, for example, chemotherapy, gemcitabine, as well as inhibitors of CA9. And what we can show together, if we inhibit both CA9 and together with gemcitabine, we have the most robust suppression of tumor outgrowth. So clearly beginning to think about these tumor microenvironments, we're not targeting the proliferative cells with the CA9 inhibitors, but we are likely targeting the proliferative cells with the chemotherapy. So I'll just leave you with this concept that there's order and heterogeneity, that the tumor cells are not random, and they're clearly organized into distinct tumor and metabolic space. There's a high concordance in the spatial distribution between our human samples and our PDXs. And of course, the challenges that we're facing now is which cells are genomically similar versus genomically different. Been fabulous people in my lab, and really Tina Grosso and Connie Ramirez has championed most of this, with fabulous collaborations with Claudia Kleiman for all of the single cell stuff. So sorry, I realize I couldn't see the time. I will stop sharing if I can. Have I done that?
Great.
Have I stopped sharing? Yeah.
Yes, you have. Thank you so much, Dr. Park. That was phenomenal. I'm even getting text messages from participants telling me how great it is. So I wish you could hear the applause you're getting. Well, let's see how far we go until we get cut off, but we have a number of questions and I'll jump right in. First, we have a question from William Beck saying, wonderful seminar, thank you. 2 questions. How do you quantitate high and low? And secondly, what turns on PD-L1 and going from immune desert to strongly restricted to fully inflamed? I guess that's assuming there is a dynamic transition.
Right. We really first quantified based on location. So these immune low are essentially negative. And you sometimes, even though you have many CD8s at the margin, you may have a few that enter at the marginal space of the tumor, but they do not enter the tumor bed. And those tumor beds are not uniformly hypoxic. We've stained them with CA9. So this is not just hypoxia. Roberto Salgado has published quite a nice paper on quantification and machine learning. So there's quite a lot of data in his paper that would give you cutoffs for counting. And PD-L1, yes, I've got no clue. I think that these are really important questions, and obviously people are using appropriate mouse models now that can let us start to look at that, and I think it's key. And of course, Nelly had some very interesting data from early stage DCIS, again showing early on that you can start to see PD-L1 positivity in some DCIS. And again, I think following these through and in tumor beds that have DCIS versus non-DCIS will be really interesting questions. Yeah.
Excellent. So another question, have you seen any difference in stem-like CD8 markers such as TCF1 between margin or strongly restricted versus fully inflamed cohorts?
Yeah, I'd have to go back and look. We did look at the time. There's no doubt that these immune cold do have signatures that are more EMT-ish, if we want to call it that. And we need to go back and look at that more carefully. We have not definitely immunostained for that. So these would be questions we'd have to ask. Yeah.
Okay, try to get through a couple more. Many human cancer single cell studies have shown single cells in UMAP are clustered by patient tumor sample, but here I see they're not. I was wondering why. Do you use some computational integration method to correct the patient effect, or is it expected for single cells from PDX?
No, I think, to be honest, I think when you've got whole patient tumors, you've got so many other cells in there that you tend to cluster based on the patient. And so what we wanted to do using the PDXs was essentially remove all of the other stuff. And we didn't count any mouse reads. Now we are looking at those mouse reads because there is stroma in these tumors. And I think that that's maybe the interesting part is that we're capturing biology, I think, because we're only looking at the tumor cells. And we know that these are not genomically different clones, although these yellow cells that can adapt, we are imagining and we're trying to test that they're epigenetically modified as they may go into these hypoxic zones. So that these will become really important questions. And of course, one would like to pull these cells back out again. And this becomes quite difficult. So I think what I really just want to leave you with this concept is that the biology is conserved between tumors, and that possibly if you were to look genomically, these tumors are not going to be identical, and even transcriptionally, globally, they won't be identical.
Okay, thank you. So unfortunately, we're out of time now, but we do have a number of questions that will be sent to you from Claudia. I have a few of my own, so I'll add those too. But again, I want to thank you, Dr. Park, for a fantastic seminar and really, inspiring all of us. Thank you so much.